This is another OCR video, A-Level Biology on Unit 5.4, Endocrine Communication. Right, so today we're going to look at the whole chapter. So we're going to look at hormones, endocrine, exocrine functions, the adrenal glands, the pancreas, which is quite packed full of information there, and then diabetes, which are two quotes from my notes that I'll explain later. So first of all, let's get straight into it with what are hormones. So hormones are chemical messengers that travel in the blood. They are used for communication around the body, so they're found in our blood circulatory systems, and there's two types of hormone. So you can have protein hormones, which are insoluble in phospholipid membranes, therefore they do not enter the cell. And an example of that are glucagon, adrenaline and insulin. They're all protein hormones. They bind to receptors on the cell plasma membrane. They never enter the actual cell themselves. Then we've got steroid hormones, which are soluble. They enter the cell nucleus and directly affect the DNA. And some examples of that are estrogen and testosterone. Then we've got these two words here, endocrine and exocrine. I put endo/exocrine, but remember that's endocrine, exocrine, two different words. I've just been lazy. So endocrine glands release hormones, so they release hormones into the blood, whereas exocrine glands do not release hormones, but they secrete products into ducts, such as salivary glands. So target cells are cells that receive an endocrine signal. A group of target cells form target tissue. So, for example, adrenaline directly affects liver. For example, insulin directly affects liver and muscle cells. Okay, so liver and muscle tissue are the target tissue. Target cells have complementary receptors on them, complementary receptors on them that are complementary to the hormone that they are detecting. So a specific cell, hepatocytes, will have a receptor for glucagon. Therefore, hepatocytes in the liver are the target cells, target tissue. If you do not have that receptor for the specific hormone that's complementary to the hormone, you will not be affected by the hormone. Hence, hormones are specific to certain cells, okay? Which is a good thing for us, as if there was no such thing as target cells and hormones affected every single cell in the body, that would cause hormones we don't want to be having an effect on certain cells to take an effect, such as like sex hormones, testosterone on liver, I don't know. Can't think of a decent example. You just don't want hormones that aren't relevant to the cell to be producing that effect, otherwise it will not be efficient. Okay, then we've got this thing, first and secondary messengers. So first messengers are our protein. This is an example for protein hormones, okay? They are the first messenger, so they bind to the cell surface membrane on the target cell, okay? They bind to a receptor which is complementary to them. Okay, so see all these links here, this whole topic, they all link together. You've got to learn all these keywords, first messenger, secondary messenger, target cells, endocrine, exocrine, protein and steroid hormones, they all link together. So first messengers bind to the cell surface membrane receptor, and then they release a secondary messenger, which here I've got an example of from my notes. So the hormone here, it binds to the receptor within the membrane. So the hormone's a protein hormone, it cannot enter the cell. It binds to this receptor, which activates a G protein, okay? This is a general example. So this G protein then activates adenyl cyclase, which is our effector molecule. Adenyl cyclase, upon activation, converts ATP to cyclic AMP. And that cyclic AMP is our secondary messenger. It's within the cell this is produced, and that can have all sorts of effects. It can open channel proteins, it can cause enzyme activation. So this is not a specific example, it's just a general example on how hormones work within cells, and we'll see later a more specific example over here, okay, for insulin. One thing to look at for steroid hormones, which is the last thing on this page, is they have a specific receptor within the cytoplasm. So our steroids hormones, they're soluble, they diffuse through the plasma membrane, and then they bind to a specific receptor in the cytoplasm. This forms a receptor steroid hormone complex which can enter the nucleus. So it can enter our nuclear envelope, and it binds to another specific receptor on the chromosomal material. So let's remember, the steroid hormone diffuses into the cell, it binds to a receptor within the cytoplasm, so within here. That forms a receptor steroid hormone complex which can enter the nucleus. It binds to another specific receptor on the chromosomal material which stimulates mRNA to code for protein function. So you can have a steroid hormone, it will enter, eventually after binding to two receptors, it will be able to code for a specific length of DNA by stimulating mRNA. So it can stimulate certain proteins to be produced. So steroid hormones can switch genes on and off, okay? Now let's look at an example of an endocrine organ, which is our adrenal glands. So they are an endocrine gland just above the kidney. So in this diagram here, we've got our kidneys, and this is where you'll see your adrenal glands, okay? They sit on top of the kidneys. They've got two distinct layers, an outer adrenal cortex and an inner adrenal medulla. So one way to remember which way around that is medulla, you can think of as middle, okay? Medulla is middle. Then obviously you just remember cortex, and that's going to be on the outside. So we've got our, in the blue here, we've got our whole adrenal gland. The inner bit here is our medulla, and outside of that is the cortex. And what this is showing here is like a small section cutting out of here, and it's got three zones. The outer zone is our zona glomerulosa, inner zone is our zona fasciculata, and our inner zone, I think I said inner zone twice, our middle zone is zona fasciculata, and our inner zone is zona reticularis. Okay, so that's closest to the medulla. Just remember which way round those go. That's what it looks like. And let's just remember that's just a small cut from our adrenal cortex. So that would continue in a whole ring around the medulla. Okay, so mineralocorticoids are in the glomerulosa, glucocorticoids produced in the fasciculata, and precursor molecules are produced in the reticularis. So we've got cortisol produced in the fasciculata and aldosterone produced in the glomerulosa, which are examples that OCR want you to learn, so it's important you do. So aldosterone and cortisol cause the stress response. So they increase sodium uptake within the kidneys. So sodium moves out of the nephron, causing there to be a high water potential in the collecting duct. Hence, more water is reabsorbed and less is put in the urine, not the urinal, which increases blood volume and pressure. Okay, so the more water that we reabsorb, the more blood pressure we're going to get, of course. More specifically, they increase fat and protein breakdown, and that is mostly done by action of cortisol. So metabolism of carbohydrates in stressful slash low blood glucose situations, cortisol is released to stimulate production of glucose from stored compounds in the liver. Okay, whereas aldosterone has a higher effect on this first bit here, okay, increase in the blood volume and pressure. So our precursor molecules released from the zona reticularis are taken up by the ovaries and testes and converted to sex hormones to develop secondary sexual characteristics and produce gametes. Okay, you don't have to know any specific example for a precursor molecule, I do not believe. The last thing to look at is the medulla, which secretes adrenaline and non-adrenaline. So adrenaline is a polar molecule which cannot pass through plasma membranes. It converts glycogen to glucose, relaxes smooth muscle, increases stroke volume heart rate, causes vasoconstriction to increase blood pressure, dilates pupils, increases muscle awareness, decreases gut action, and causes body hair to stand erect. So that is the hormone that initiates the fight or flight response, okay? So one way to remember what the adrenal glands do is to remember, in general, they cause stress responses. Okay, every hormone here we're dealing with is stress responses apart from zona reticularis, which putting your body in a ready state for action. Okay, hence like dilated pupils, so you're more aware of your surroundings. Muscle awareness decreases gut action because it's not really necessary. Increase in the heart rate, so you're ready for action. Okay, probably the most well-known hormone is adrenaline. Okay, now the pancreas, and this page looks horrible, but I wanted to fit it all in one to summarise it, and this is what I'm doing. I'm making these videos to revise myself, and it's nice that if people do like to watch them. Sorry if I'm quite slow at speaking, but I'm putting myself first. I'm taking my level in like 4 weeks' time now, so I'm just making as many of these videos as I can because they really do help me learn by teaching it to other people. But I hope you're enjoying them too. So the pancreas has two functions, endocrine and exocrine. Endocrine function, as we know, is hormones. Exocrine is stuff that's not hormones, basically. So let's just look at its exocrine function first. So it creates these things called pancreatic juices. So pancreatic amylase, trypsinogen, lipsin, lipase, sodium hydrogen carbonate, which neutralizes some stomach acid, which you'll realize why in a second. And as you can see, these are all digestive enzymes. So pancreatic juices are secreted into the small intestine. So that's just after the stomach. So this sodium hydrogen carbonate neutralizes that stomach acid for these enzymes to be able to function. So how does this exocrine function work then? So it's got these cells called achenes, or achene. A small group of cells surrounding tubules are called an achenes. Achene are grouped together in small lobules separated by connective tissue. They secrete digestive enzymes into tubules. Okay, and these are our digestive enzymes. These four here, that's not an enzyme. Tubules then join together to form intralobular ducts, and then all these intralobular ducts go into a pancreatic duct, which enters the duodenum. I wasn't going to do this, but to sort of describe what we're looking at here. Actually, here's the diagram. So an electron micrograph, or a light micrograph, probably is light, it doesn't look very detailed. Here is an achene, so it's a group of achenes separated by connective tissue, and then it's hard to see, you can't really see the tubules here. You might, just above there, that hole there, it could be the tubules that join together to form intralobular ducts, and then all the tubules, so the tubules from different achene join together, they form intralobular ducts, which go into one large pancreatic duct into the duodenum, which is within the small intestine. Okay, so that is the exocrine function. You might want to look at achene and look at a few more diagrams yourself. I couldn't fit too many diagrams on this page, it was really packed, because I realised I'm not really explaining that too well. Anyway, we've got the endocrine function, which is the whole point of this topic. So hormones. Hormones are produced in the islets of Langerhans, which is this area here. This is the islets of Langerhans. So within this islet of Langerhans, we've got alpha cells and beta cells. You won't be able to differentiate from a graph like this, sorry, a micrograph like this. But within each islet of Langerhans, we've got lots of alpha cells and lots of beta cells. So alpha cells secrete glucagon. So one way to remember this, by the way, glucagon, so it's got an A in it, alpha. Beta cells secrete insulin, there's no A in insulin. So you can just remember glucagon, alpha, beta will be insulin because it's not got an A in it. That's how I remember it anyway, and it works. So glucagon impacts liver cells. It activates enzymes within the liver cells which convert glycogen to glucose, which is known as glycogenolysis. Okay. By the way, there's three long words you're about to come across, and you've just got to, it's just one of those things you've just got to learn them again and what each of them are. Glycogenolysis. So that's conversion of glycogen to glucose. Gluconeogenesis is another thing that's activated by these enzymes, and that is the conversion of fatty acids and amino acids into glucose. That's gluconeogenesis, okay? And also lowers the rate of respiration of glucose, which I've not written. It lowers the rate of respiration of glucose, because glucagon, of course, is released in situations where we've got a low blood glucose, so we need as much glucose to be produced as possible. So we go to our storage and try and convert things to glucose, because it's important for lots of cells to function. Insulin is released when we've got a high blood glucose and we need to bring it down. So insulin affects liver cells and muscle cells. Remember, liver cells can be called hepatocytes. So if you see hepatocytes in the exam, just know that's a liver cell, which I might do a video on in the future. They increase cell permeability to glucose. They activate enzymes to convert glucose to glycogen, so that's the opposite of glycogenolysis, and this is called glycogenesis. This is what I mean. The words are so similar, it's really hard to remember them. I've not figured a way to remember them yet. I'm going to. I think you've just got to keep telling yourself, so glucose to glycogen is glycogenesis, glycogen to glucose is glycogenolysis. Okay, long words, hard to remember. The final thing that insulin does is increases the rate of respiration of glucose. So all these things decrease the amount of glucose we have in our blood. So how is insulin secreted? So I've got 7 steps here. So we've got potassium channels within beta cells of these little Langerhans. So we've usually got potassium. I've got this potassium channel in red. Usually potassium is flowing out freely through these potassium channels. So it's usually flowing out either way, and potassium is just leaving the beta cells. Let's get rid of those arrows now. What happens is, in high blood glucose, glucose enters the beta cells. So this thing here is a glucose molecule. I'll put a G in it so you can remember that. This is glucose. It's not very clear for you. Glucose. Okay. So high blood glucose, glucose enters the beta cell. This glucose is respired to produce ATP. So it's converted to ATP. You can watch my respiration video on how that happens. It's not that simple, because it takes me about 27 minutes to explain it to you. But if you know what respiration is, all you have to know, glucose is converted to ATP eventually. And then this ATP closes these red potassium channels, okay? So potassium now cannot flow out of the cell. So potassium, which is K+, they can accumulate inside. So this accumulation of positive potassium causes the cell to become less negative. This is depolarization of the cell. So accumulation of potassium depolarizes the cell. The depolarization opens these yellow calcium channels. And I've just realized I've put sodium. So let's rub that out. Rub that in, out. Because that's not very helpful for anyone. They open calcium channels. Okay. The calcium then enters the cell causing insulin vesicles, which I've got here, so the black bit's the vesicle. Inside I've got insulin to fuse with the plasma membrane, releasing insulin by exocytosis. So here we've got our cell, calcium enters, the presence of calcium causes these insulin-containing vesicles to fuse with the cell membrane. When they do fuse it, by exocytosis they release the insulin into our blood vessel. The final thing to look at is how insulin then impacts a cell. So we've seen it's just been secreted into the blood. So imagine the bloodstream goes over here where my arrow is, and then it releases the insulin molecule at the target cell, which will be in the liver or the muscle. Okay, liver and muscle. I've pointed a glucagon there. I've got so much information on here. So as we can see, glucose is sat outside the cell because we've got a high blood glucose concentration. We can picture this bit here to be our blood vessel. We've got glucose in the blood vessel. There's too much of it. Insulin's been secreted into the blood by the beta cells. It then binds to a receptor specific to insulin. So a receptor on liver or muscle cells binds to this insulin. this activates this enzyme here. Hormone binds the receptor, activates the G protein. In this case, we've got insulin bind to the receptor, which activates the enzyme tyrosine kinase, which replaces our G protein. Okay, this causes inactive enzymes to become active enzymes. So our tyrosine kinase, if we go back again, is our effector molecule. Okay, tyrosine kinase then, so inactive enzymes into active enzymes, which are drawn like a wiggly orange bright line around it to show it's been activated. And these active enzymes cause an enzyme cascade, which causes these glucose transporter proteins bound within vesicles to fuse with the plasma membrane. So what happens here then, if I get rid of this enzyme cascade causes the fusion of, I'm really bad at speaking and doing something at the same time, it causes the fusion of these glucose transporter proteins into the plasma membrane. Okay, so what we've seen here, this vesicle's bound to here, it's embedded the glucose transporter protein into the membrane, allowing glucose to enter the cell. So if we get rid of this, let's make that a bit bigger so I can delete it quicker. So now we've got our glucose transporter protein in our membrane here. This allows our glucose molecule to enter the cell, which will be our liver or muscle cell. It enters the cell. This glucose is converted to glycogen. So glycogenesis happens once the glucose is in here. So let's draw an arrow. All these glucose molecules then bind together. Select that. Copy it. I'm now creating a glycogen molecule. This is not what it looks like, but it's just one way to show it to you. Okay, so the glucose molecules are bound together to form glycogen, okay? Which looks a bit like honey there, honeycomb. Right, that was the pancreas. That is all you need to know about the pancreas. But there's one big word that I've not spoken about, it's negative feedback. So glucagon and insulin work through negative feedback. I've not done a video on unit 5.1 which will explain negative feedback in detail, but basically negative feedback causes the opposite to what's been detected. So if you've got low blood glucose, really low blood glucose, we release glucagon to get high blood glucose. So we want the negative feedback is creating the opposite of what we've got. So if we've got high blood glucose, that's bad, and we want to lower it. So we detect high blood glucose, and then we release insulin to create a lower blood glucose. So that's negative feedback, and that's its role in the pancreas. So it's a bit of homework I had to hand in, but it's a really good table to summarise it. The comparisons between type 1 and type 2 diabetes, and then their treatments, which I'll go through in a bit more detail on the next page. So type 1 diabetes is caused by damaged beta cells, so insulin isn't produced, whereas type 2 diabetes is caused by a lack of responsiveness to insulin. So the receptors on the surface of the liver and muscle cells do not any longer respond to insulin. So insulin is still produced in type 2 diabetes, but we can't respond to it. And this is usually caused by too much insulin being produced over your lifetime because of a high sugar diet, obesity, lack of regular exercise, aging, okay, Afro-Caribbean origin, all that sort of business. But type 1 diabetes is less common, and it's caused by an autoimmune response to attacking its own beta cells or a viral attack on beta cells. So type 1, you can't produce insulin. Type 2, you can't respond to insulin. And these are our differences. So type 1, you suffer mainly from a low blood glucose levels. You might be thinking, why is that? If you can't produce insulin, surely you have high blood glucose. Well, that is because insulin converts glucose to glycogen. So if you can't produce insulin, and you use up all your blood glucose, you do not have any backup stores in glycogen. So you don't have any glycogen. If you use up all your blood glucose, you have a period of hypoglycemia, lack of really low blood glucose, which can be dangerous, and it causes the following symptoms like thirst, urination, bedwetting, extreme hunger, which you don't really need to know too much about, I don't think, which is part of my homework. Whereas in type 2 diabetes, you suffer mainly from high blood glucose levels because we're not responding to glucose. Here are our treatments, okay? So we have insulin injections, which is monitoring the blood glucose constantly to see if they're at the correct levels. So diabetes 1, in a way, is easier to control. You just inject yourself with insulin as you can't produce it, and you can still react to it. Second one is insulin pump therapy, which is a permanently inserted needle under the skin, constantly pumps insulin at a controlled rate into the bloodstream. So you just top that up daily, I think, and it does it itself. You don't have to inject yourself every single time. Islet cell transplantation, so you transplant healthy cells from a deceased pancreas donor, and you get your beta cells, and then they function, and you can produce insulin. Or you can have a whole pancreas transplant, okay, which is probably slightly more common than just the islet cell transplantation. For type 2 diabetes, you can change your lifestyle, because we said it's caused mainly by obesity. If you regularly exercise, avoid high sugar, then you've got these two things here. So metformin improves the sensitivity of your body tissues to insulin. So that's a drug. It also lowers glucose production in the liver. And bariatric surgery is used for obese people to reduce the size of their stomach with a gastric band to encourage weight loss, which is seriously life-threatening, and they only do it for really serious situations. Here is another image in my notes talking about, it's talking about insulin production. I'm not going to spend too long on this page because I realise that you might not be able to read it. I've spoken to you about all of these methods though. If you can read it, excellent, you can pause the video and have a read of that yourself, but I'm not going to speak about it again. Okay, so I hope you enjoyed this video. More videos come. You can watch my previous videos as well. But once again, I'm making these videos for myself really, because I'm taking the exam soon. I don't have enough storage space on my iPad, but in a way it's really nice that if people do watch my videos, I don't think many people do though. You might start to see a few more faces in the future. Alright, cheers.